What if one single HIV virus entered your blood? Just one, not a drop of blood, not a needle, not millions of viruses. One tiny particle far too small to see floating into the bloodstream of a healthy person. Would that person instantly become infected? The honest answer is almost certainly not. But the real science is much more interesting than a simple yes or no, because infection is not a magic spell. It is not like flipping a light switch. It is more like a tiny invader trying to break into a giant castle. And that one lonely invader has to pass many locked gates before anything serious can happen. First, the virus has to still be infectious. That word infectious simply means still able to start an infection. A virus is not a tiny animal with a brain. It does not think, it does not hunt, it does not plan its attack. A virus is more like a tiny package of genetic instructions, but if that package is broken, it cannot do anything. Imagine a key snapped in half. It may still look like a key, but it cannot open the door. A damaged HIV particle may still look scary in a drawing. But if its outer shell or surface proteins are damaged, it cannot successfully infect a cell. So the first question is not simply, did HIV enter the body? The first question is this virus still intact and working? If the answer is no, the story ends immediately. Now suppose the virus is still infectious. It enters the bloodstream. Now it has a second problem. It has to find the right target. Your bloodstream is enormous compared with one virus. Imagine dropping one paper boat into a huge river system and expecting it to reach one specific dock. That is the situation for one lonely virus. It cannot infect just anything it touches. It is not looking for red blood cells. It is not looking for random tissue. HIV mainly targets certain immune cells, especially CD4 T cells. A CD4 T cell is a type of white blood cell. White blood cells are part of your immune system, which is your body's defense force. CD4 T cells are like commanders in that defense force. They help organize the immune response when your body faces infections. That is why HIV is dangerous. It attacks part of the system that helps coordinate your defenses. But one virus still has to find one of these suitable target cells. If it simply drifts through the bloodstream and never meets the right cell, nothing happens. The invader gets lost before reaching the castle gate. Now imagine it does reach the right kind of cell. It still cannot just walk in. Cells are not open houses. They have surfaces, barriers, and molecular locks. HIV uses special proteins on its surface to attach to certain receptors on the surface of a target cell. A receptor is like a tiny door scanner. The virus has something like a fake key card. If the fake key card does not fit the scanner, the door stays closed. If the fit is good enough, the virus may enter. But even this is not guaranteed. Biology is messy. Molecules bump around. Shapes have to match. Timing matters. The local environment matters. One virus has no backup team. If this one attempt fails, there may be no second try. Now, let us say the virus somehow enters a CD4 T cell. That still does not mean the person is infected. Now comes the next challenge. The virus has to hijack the cell. HIV carries its genetic instructions as RNA. RNA is a kind of biological message. Human cells usually store long-term instructions as DNA. DNA is like the main instruction library inside the cell, so HIV has to do something clever. It must turn its RNA instructions into DNA. Then it tries to insert that new viral DNA into the cell's own genetic library. That sounds complicated, so here's the simple metaphor. Imagine the virus sneaks into a factory. The factory already has a giant instruction manual. The virus tries to slip in a new page. If that page gets accepted, the factory may start making virus parts. But again, this is not automatic. The copying step can fail. The viral instructions can be damaged. The cell can detect something is wrong. The cell can shut down. The infected cell can die before making new virus. So even after entry, the virus still has to win several more rounds. One virus has to succeed again and again. Now suppose it wins even that step. One cell becomes infected. Is that enough? Not necessarily. One infected cell is not the same as a full infection in the body. For an infection to become established, the virus has to spread. That infected cell must make new virus copies. Those new viruses must leave the cell. Then they must infect more cells. Then those cells must make even more viruses. This is how a tiny beginning can become a growing infection. But with only one virus, every failure matters. One mistake can break the whole chain. This is why dose matters. Dose means how much infectious virus is present. One spark is not the same as a wildfire. One spark can start a fire, but only if it lands on the right material, avoids being blown out, and finds enough fuel. Most sparks disappear. One lottery ticket is not the same as a million lottery tickets. The more tickets you have, the more chances you get. The more infectious virus is present, the more chances the virus has to pass all those gates. One virus gives it almost no room for failure. That is why real HIV risk is not usually discussed as one virus. In real life, risk depends on several things: how much virus is present, what body fluid is involved, how the fluid enters the body, whether there is access to vulnerable tissue or bloodstream, whether the person living with HIV has a high viral load or an undetectable viral load. Let us define viral load. Viral load means the amount of virus in a person's body fluid or blood. High viral load means there are many virus particles. More virus means more chances. Low viral load means fewer chances. Undetectable viral load means treatment has pushed the amount of virus so low that standard tests cannot detect it. And this brings us to one of the most important modern HIV facts. U equals U undetectable equals untransmittable. That means a person living with HIV who takes effective treatment and keeps an undetectable viral load does not sexually transmit HIV. This matters because HIV is not a curse. It is not a punishment. It is a virus. And science has changed the story. A person living with HIV can take treatment, stay healthy, live a long life, have relationships, have a family, and when their viral load stays undetectable, they do not sexually transmit the virus. Now let us also talk about what HIV does not do. HIV does not spread through air. It does not spread through hugging. It does not spread through hugging. It does not spread through shaking hands. It does not spread through sharing food. It does not spread through toilet seats. It does not spread through casual touch. HIV needs specific body fluids and a real route into the body. Those body fluids include blood, semen, vaginal fluids, rectal fluids, and breast milk. The route matters. A virus sitting outside the body is very different from virus reaching vulnerable tissue or direct blood access. This is why contact and infection are not the same thing. Exposure means the body may have encountered a virus. Infection means the virus successfully entered cells, copied itself, spread, and established itself. Those are very different. Think of it like someone trying to break into a castle. Exposure is seeing someone outside the castle wall. Infection is when that person gets through the wall, reaches the control room, takes over the system, and starts bringing in more invaders. A lot has to go right for the invader, and a lot has to go wrong for the castle. Now, this video is a thought experiment, but real life still matters. If someone has a real possible HIV exposure, there is emergency medicine called PEP. PEP stands for post-exposure prophylaxis. That sounds complicated, so break it down. Post means after, exposure means possible contact with the virus. Prophylaxis means prevention, so PEP means prevention medicine after a possible exposure. It is time sensitive. It usually needs to be started within 72 hours. The sooner it starts, the better. So the lesson is not panic. The lesson is understand the route, understand the risk, and act quickly when there is a real exposure. But do not fear impossible everyday contact. So, back to the original question. What if one single HIV virus entered your blood? It would not be an instant infection spell. That one virus would need to survive. It would need to find the right cell. It would need to attach. It would need to enter. It would need to copy itself. It would need to insert its instructions. It would need to make new viruses. It would need to spread before being stopped. One failure breaks the chain. So yes, in theory, one infectious virus particle might be able to start an infection, but in real life, that would be extremely unlikely. The real world is not one virus equals infection. The real world is dose, route, viral load, timing, and biology. Infection is a process, not magic, not a curse, not a light switch. One tiny virus is not an all-powerful spell. It is one fragile invader facing many locked gates, and most lonely invaders never make it through. The scary question gets smaller when you understand the science, and that is the real point. Fear says one virus means doom. Science says, let's look at the process. And when you understand the process, you can see the real size of the risk.